In this video, I'll be going over central nervous system and neurophysiology, and here's a little bit of an outline of what I'll be going over specifically. So I'll first start by talking about brain areas and functions. This is going to be pretty quick. And then I'll get into the three different types of channels that we talked about during Unit 1 of this class. And then I'll end with a graded and an action potential, what types of channels are where, and what's going on with the whole neuron during this process. So let's start with some brain areas. And you'll probably notice for pretty much this whole discussion, I'm not really going to be doing the discussion worksheet that is for you on Canvas. Instead, I've kind of just prepared this material. This material is, though, very similar to your discussion worksheet. So after doing this, you should be able to do the discussion worksheet, at least everything that I've covered in here. The reason why I'm not working directly off of the discussion worksheet is because I've tried to make things more streamlined for what you need to know, and I think the discussion worksheet can go just a little bit further at some points. So I've pulled kind of the most important parts, at least what I consider to be the most important parts from the discussion worksheet, and that's what I'll be doing in this video. So the first thing I want to start off with is brain areas, which is the last part of the worksheet. But for this, what I want us to do is to try to really just put like a 1 to 2 word function for each of these structures, so that if we were to be able to match them to their function, then we could do that easily. So that's what we'll do here. So we'll start with the hypothalamus. The function of the hypothalamus is mainly homeostasis. It does have the endocrine functions that we talked about, and yes, that's true, but in terms of the central nervous system functions, it mainly regulates homeostasis, things like thirst, hunger, osmolarity, body temperature, stuff like that. Your thalamus is a sensory conductor. And I'll kind of put conductor in quotation marks. So I put sensory conductor because what the thalamus does is it gets sensory information from your 5 senses, technically 4 because your olfactory does not go through the thalamus. So it gets information from 4 of your senses other than smell, and it sends that information initially to the thalamus, and then the thalamus kind of reroutes those signals to the respective part of your brain that will then process it. So your thalamus will first receive something like the visual information before getting sent to the occipital lobe. Stuff like your auditory information will first go through the thalamus before going to its respective lobe. So that's what happens with the thalamus. It's kind of a conductor, kind of gathers the sensory information in one place and then directs it on where to go to the brain. The hippocampus is for long-term memory. So this was the example, or one of the examples, that Dr. Chang talked about in class. The hippocampus is responsible for the consolidation of short-term to long-term memories. So without the hippocampus, you can no longer form new long-term memories. So you would forget things once they leave your short-term memory. So hippocampus is consolidation of memory. Basal nuclei is motor functions. I'm going to put in parentheses kind of more fine motor functions. So the basal nuclei, the reason why I put fine motor functions is because the cerebellum is also motor functions. But I'll put in parentheses here gross. So the cerebellum, it is also working in motor functions, but it's kind of more gross or larger movements. Things like your posture, your gait, your balance, whereas the basal nuclei tends to kind of smooth out more fine motor functions, kind of more dexterous things that you may think about using like your fingers, stuff like that, or your hands, whereas the cerebellum is more kind of full body motor functions. Okay, prefrontal cortex is for high level reasoning. And or decision making, whichever way you want to think about it, high level reasoning or decision making. We talked about in class that this is kind of what like makes us human. It kind of gives us the ability to process and make decisions. The brainstem, the medulla, and the pons is your mainly regulating your autonomic nervous system. This regulates things such as your blood pressure, your heart rate, your respiratory rate, these automatic functions that occur in your body that you don't need to think about. So that comes from the medulla and the pons, and we'll talk more about that in the cardiovascular system. The limbic system is for emotions and memory. I'll kind of put memory here with a star because I don't want you to think it's really related to the hippocampus in terms of like the storage, but rather it ties certain memories to certain emotions that you've had at certain times. If you want to talk about in office hours, there's a really interesting episode of Black Mirror that really plays around with the physiology or the function of the limbic system. So yeah, if you're wondering what's going on there, ask me in office hours. But yes, these are the kind of 1 to 2 word functions that you can relate to the brain structures. Okay, now let's get into some neurophysiology. And I want to start by talking very generally about the 3 types of channels that we have in our body. And those 3 types are ligand-gated, voltage-gated, and mechano-gated. So what I've done here is I've drawn what is supposed to be a channel in a cell membrane, and on the left they're all closed and on the right they're all open. So let's kind of talk through each one and talk about, well, what makes them open. So the thing about channels, and this is a difference from a carrier, the thing about channels is that they have gates on them. And just like a gate in real life, in order to open a gate you need to unlock it. In the case of these channels, there's 3 different types of gates that require 3 different types of keys, if you will, to unlock them. And the first type of gate is a ligand gated, and this would be most analogous to like a literal lock and key to open a gate. So what's happening here on the left is we have this closed channel, and in green I've put a little receptor here. That would be for the ligand to bind to. So let's say we have some sodium ions out here. So let's say this is a ligand-gated sodium channel. Well, as of right now, this sodium cannot pass through the cell membrane because the channel is closed. It hasn't been unlocked yet. But let's say a little neurotransmitter is floating out here and it can make its way to that receptor. So what happens when the neurotransmitter binds to that receptor? Well, once the neurotransmitter binds to that receptor, the neurotransmitter can be referred to as a ligand. You might also hear this referred to as a chemically gated channel. They're the same thing. It's just saying this chemical or this molecule, this ligand, is binding to the gate. So when the ligand binds to it, that channel opens, and now the sodium can go in. So that would be a ligand-gated channel. These are mainly found on dendrites and cell bodies of neurons. And they're most often used to trigger graded potentials. So these are really important for graded potentials. All right, let's go down to voltage gated. Now this is where it starts to get a little bit more abstract. It kind of goes further away from the lock and key analogy of a gate, but it's still a gate nonetheless. But this time the key to open it is a change in voltage. So if let's just say like outside is 0 millivolts and inside is minus 70 millivolts, well there would need to be some sort of change in this difference in voltage. So maybe the inside goes to, let's say the inside somehow goes to minus 50. Well, then that change in voltage inside the cell will trigger the channel to open. And we can talk later about what's actually making this jump happen. So this change in voltage So change in voltage is the key to opening this gate. So if we have sodiums outside initially, we say they can't get in. But once we have a change in voltage, well, now the sodium can just go right into the cell, which would further make the voltage change. So if it was like initially minus 50 when the sodium started entering, well, then that minus 50 would maybe go to something like positive 30 millivolts because more sodium is entering. But you need that initial intracellular change in voltage in order to trigger the opening of that channel. All right, the last one is the mechanogated channel, and instead of referring to like the key opening a gate, this would be like if you just physically forced your way through the gate, like you knocked the gate down or pushed it to the side entirely. But if you can kind of imagine a cell membrane being pushed to either side, being physically stretched open, well, that physical force of stretching the cell membrane will just physically open the gate. So if we have sodium out here, not able to get in, but after this physical stretch, that channel will open and sodium can move in. All right, so the voltage gated channel, this is mainly found in the axon and also the axon terminal. So the axon and axon terminal is mainly responsible for action potentials. So these voltage-gated channels, super important for action potentials. Mechanogated channels, we're not going to really look at them in the example I'll talk about today, but mechanogated channels often found in places like skeletal muscle where the tissue is changing shapes quite often because of contraction and relaxation. So these sensory fibers, sensory nerves that are innervating muscles usually have mechanogated channels on their nerve endings because they are activated when the muscle contracts and relaxes. But we will definitely talk about mechanogated channels later on in this class. But for now, let's just kind of focus on voltage and ligand, and I hope this gave you a better idea of just generally what's going on. Okay, so now let's look at an action potential. So what I've done here is I have kind of copy and pasted two drawings of a neuron together. So if you want to, find drawings of neurons to copy and paste together, or just quickly draw your own neurons. And then also down here, if you also would draw an axis for yourself and label it the way that I have labeled it so far. Because what I'm going to do is we're going to basically go along this drawing and show at every part on the neuron what type of channel is being activated, and how would that look if you are recording that neuron electrically using electrodes. So if you want to just kind of pause the video and draw this out to get ready for the notes that we are about to take. Okay, so let's start up here with the cell body. So like I said, this is going to be the example of if we were able to place electrodes at certain parts of the neuron and then able to measure the change in voltage that's happening. So I'm just going to erase that because we assume that that's what we're able to do here. So we're going to start down here on the graph. I'm just going to start by drawing a flat line here. The resting membrane potential of a neuron, we say, is around minus 70 millivolts. So I'll just draw the flat line there. And that is just saying nothing is happening to the neuron right now. So the neuron is at rest, not getting any signals. But let's say, so if you could imagine like another neuron over here, before this one, releases a bunch of neurotransmitter into this synapse. So what this neurotransmitter is going to do is it's going to bind to a bunch of ligand-gated ion channels. So right here in the cell body and the dendrites, we have ligand-gated ion channels. And these can be really any kind of ion channels. Most often you'll hear us talk about ligand-gated sodium channels, but they can be ligand-gated potassium chloride, or even calcium. But you'll most often just kind of hear ligand-gated sodium channels in this class, and ligand-gated chloride channels. Now I forget what I said. You'll most often hear us talking about ligand-gated sodium channels in this class for excitatory signals, and ligand-gated chloride channels for inhibitory signals. Okay, so in this case, let's just say it's a ligand-gated sodium channel. So when that happens, a bunch of extracellular sodium now has the ability to enter the dendrites in the cell body, making this cell body become more positive. So what happens here is we get a little jump up in membrane potential. It just becomes a little bit more positive. So if we keep having these neurotransmitters stimulate ligand-gated or open ligand-gated channels, you'll have more and more sodium coming in. And the way this looks on this graph is you just keep kind of jumping up towards membrane threshold. So all of these individual little jump ups, the little jumps in membrane potential, is called an EPSP, an excitatory postsynaptic potential. So if this were the synapse here, well, this neuron is post-synapse because it's after the synapse, and it's an excitatory signal because it's becoming more positive. So EPSP, and this is due to ligand-gated sodium channels. All right, so once it reaches membrane threshold, now we get an action potential. So once it reaches here, we will jump up very quickly to positive 30. And on this neuron, the blue here would be happening right here at the axon hillock. So that would be considering the case of whatever stimulus started this sequence of events was strong enough to trigger an action potential. So if we consider that, we'll say, okay, even though the graded potential can decay, it's still going to be a strong enough signal to cause an action potential. So right here we get opening of voltage-gated sodium channels. And when we are able to record that on this graph, you get a very quick spike up in membrane potential. Because there's such a high concentration of sodium around the neuron, and there's a high density of voltage-gated sodium channels, when these channels open, sodium very, very quickly rushes in and it depolarizes the cell. So this movement, when you're getting more and more positive, is called depolarization. And it is due to, like I said above, voltage-gated sodium channels opening, where sodium moves in. Okay, so along the entire axon, you will get the opening of these voltage-gated channels. And this axon in particular has myelin. So in this case of a myelinated axon, the type of conduction that our action potential would be a saltatory conduction, or a jumping conduction, because what the myelin does is it physically blocks space on the cell membrane of the neuron. And if the myelin is wrapped around the axon, well, wherever the myelin is, no channels can open there. So instead of needing to open along the entire length of the axon, the channels are only going to open at the nodes of Ranvier. So if it starts at the axon hillock, it's going to jump to this node, then jump to this node, and it's going to keep jumping. So what happens is you get a very, very quick depolarization and a very quick action potential. So we do have both myelinated and unmyelinated neurons in our body, and the myelinated ones have a faster conduction speed than the unmyelinated ones. Because of this jumping, they don't need to open channels and move sodium across the entire length of the axon. They only need to do it at certain points where the axon is exposed. So that is the process of depolarization. So after sodium channels open across the entire length of the axon and sodium moves in, very quickly they will close. and what will happen is potassium will start to move out. Now, I know I'm drawing potassium being kind of on top of the myelin. That's not what would actually happen. The potassium would only be able to leave also at the nodes, but just, kind of teaching purposes and simplicity here, I don't have enough room on this axon to show everything that's happening at the nodes. So I'm just simplifying here and saying potassium is leaving. But just keep in mind, this would actually only happen at the nodes. So potassium opens, and this is called the repolarization. So it'll repolarize, and those channels are voltage-gated potassium channels that are opening. So I'm going to put a dot here, because right at this dot, what's happening is voltage-gated potassium open, while simultaneously voltage-gated sodium close. So if we're able to record, again, the potential across the entire axon, what we would see is a very quick drop in membrane potential. And this, again, is called repolarization. And if you're interpreting this graph, we're kind of moving left to right. So I'm drawing arrows on this to show you which direction we're reading this. So the repolarization is due to the voltage-gated potassium channels opening, where potassium moves out. All right, potassium will keep leaving the cell until it reaches about -90, which is about potassium's equilibrium potential, and then it will finally be brought back up to resting membrane potential. So down here, this is called hyperpolarization, or hyperpolarize. So I don't have anywhere on the actual neuron to show the hyperpolarization, but while recording it, this would be kind of like at the very end of the action potential once things are resetting and getting ready for the next action potential. But down here where it dips below the normal resting membrane potential of -70, you have the hyperpolarization. Okay, so we're done with really the graph here, at least for now, but let's come back up here and finish the series of events on the axon. So we have depolarized and repolarized the axon. So the action potential has traveled the length of the axon. So now let's get down here to the axon terminals. And this part isn't shown on the graph, that's why I'm only going to be focusing on this drawing. So down here at the axon terminals, we have a change in voltage, because we got a lot of sodium coming in from the axon. So we have a large influx of positive ions, and that will open. So on here, that influx of sodium ions causes a change in voltage that opens voltage-gated calcium channels. So voltage-gated calcium channels will open, and even more calcium will flood into the cell. Now, calcium's role is to trigger exocytosis of neurotransmitters. So once calcium enters the cell, we get release. It's supposed to be a different color. We get release of a bunch of neurotransmitters into the synapse. Again, this gap is called synapse. So the role of the neurotransmitters is to go and bind to a bunch of ligand-gated sodium channels, and then we will just repeat this process again. Ligand-gated sodium channels will open, sodium floods in, causes an action potential, depolarize, repolarize, and then neurotransmitter release at the end. So I think it's really important to be able to go over this graph on your own and say at every single part of the neuron, what channel is responsible for that portion. So in the cell body and the dendrites, it's a ligand-gated sodium channel. In the axon, it is the voltage-gated sodium channel depolarization and a voltage-gated potassium channel repolarization. And in the axon terminal, it's the voltage-gated calcium channel neurotransmitter release. Okay, so that's all I had for the drawing, but let's come down here and do one more thing on this graph. So if we were to think about the next action potential that happens, before I was showing the EPSPs, it's just kind of jumping up in potential. And that is happening, but what's actually happening is we do have EPSPs, but we also have some IPSPs. And what happens with a graded potential is the sum of all EPSPs and IPSPs. So the ones that are going down here, the ones that are going down in membrane potential are IPSPs. Inhibitory postsynaptic potential, and that would be something like a, not voltage gated, this would be something that comes from a ligand gated chloride channel. And the neurotransmitter GABA is what usually opens the ligand-gated chloride channels, so it makes the cell body more negative. So what a graded potential is the summation of all EPSPs and IPSPs. And if the signal is strong enough to pass membrane threshold, well then that initial stimulus would be a supra-threshold stimulus. And if you were to sum all of the graded potentials and all of the EPSPs and IPSPs, and you do not reach membrane threshold, then the initial stimulus is called a sub-threshold stimulus. So I just wanted to remind you of the EPSP and IPSP and what's actually going on, because when we're usually doing these graphs, we usually just kind of show the immediate depolarization from resting membrane potential, but that's not what's actually happening. You have a series or a combination of both excitatory and inhibitory, and it's the summation of all the signals that creates an action potential. Okay, so I know this video went a little longer than usual. It's almost 30 minutes here, but this is the only video, so only one part to the CNS and neurophysiology discussion. But I hope this helped. I definitely recommend being able to go through this again on your own before exam 2.